Dear everyone, my name is Liu Jinhuang. I will introduce the paper published on iMeta. The title is Microbial Network Signatures of Early Colonizers in Infants with Eczema. Eczema, also known as atopic dermatitis, is an immune-related inflammatory skin disease that often develops early in life and continues into adulthood. Establishment of gastrointestinal microbiota during infancy affects immune system development. Perturbations in the microbiome during this period can contribute to the development of immune-mediated diseases. Figure 1 Panel A shows the flow scheme of the study population. From June to October 2021, all the pregnant women admitted to the obstetric department of Zhujiang Hospital in Guangzhou, China, were assessed for eligibility. Birth mode, maternal antibiotic use, and breastfeeding ranked the top influential factors in microbiota development, and we accordingly designed strict inclusion and exclusion criteria to minimize the bias. As a result, a total of 34 mother-infant pairs were included in the current study. The result of the routine examinations of mother-infant pairs are shown in Table 1. It can be seen that the maternal pre-delivery BMI of the case group was significantly lower than that of the control group, and mothers in the case group had significantly higher triglyceride levels before delivery. Later in the research, we accordingly performed further analysis on the factors differed between groups. The step-up graphs, inverse Simpson indexes, and principal coordinate analysis show that, comparing with the microbial structure at the meconium stage, the structure of the gut microbiota at first, second, and third months was significantly different from that of the meconium. The result also suggests a gradual changes in the gut microbiota in early life, and the gut microbiota richness increases are more significant in the control group infants. We constructed time series molecular ecological networks. The results were further collaborated by randomly selecting 16 healthy infants from the Swedish cohort in the study of Roswell and the Danish cohort in the study of Christensen, respectively. We demonstrated that the network topological indexes were at a high level in samples taken from newborns, and the increased trend can be seen along with the maturity of the gut microbiota. By comparing the network structure between healthy infants and infants with eczema, we found that healthy infants adopt more complex and stable network structure in the gut microbiota than that of the infants with eczema. To probe the development of early colonizers in the infant gut, ternary diagrams were applied to represent biomass conversion processes. From the ternary diagrams, we can also conclude that the development patterns of the gut microbiota are quite different between infants with and without eczema. In the present study, we observed that the relative abundance of lactobacilli in the infant's feces decreased significantly after the initiation of breastfeeding. In contrast, the bifidobacilli were significantly higher in gut by the onset of diet. In order to identify the variables independently linked with the occurrence of eczema, variables with p-value lower than 0.05 in the univariate analysis were chosen as potential independent variables for the multivariable logistic regression. From the result, high maternal triglyceride level in the late pregnancy was identified as a risk factor for eczema. In our current investigation, our data demonstrated that healthy infants from different ethnicities share similar gut microbial network changes in the early life, while infants with eczema present a less complex and stable network compared with healthy infants. Using longitudinal data, we demonstrated that the microbial structure of infants with eczema was aberrant in the third month. The findings also emphasize the potential protective factor inverse Simpson index of the gut microbiota and the risk factor high maternal triglyceride level in the late pregnancy may be correlated with the onset of infantile eczema. This is the end of the presentation. Thank you all for listening.